AI is all the craze right now. I feel like every time I get on the internet, I interact with AI in some way, shape, or form. Be it egregiously fake AI images to using ChatGPT to bounce ideas off of, AI is seeping into everything. Along with the advent of AI, being so integrated are the inevitable discussions of our future and how AI will shape it. This talk of AI has been a hot conversation for a good number of reasons. On one hand, people can't help but remember the Terminator movies or HAL from 2001. It seems like for the first time we're truly getting close to artificial intelligence, and that can be scary. On the flip side, AI may also lead us into a new chapter of human history where many of the problems that have plagued humanity for years are solved. One such area of intrigue and possibility is how AI is shaping biology, and more specifically, protein folding. AI has made quantum leaps forward in this field, to say the least. Leaps that hold the promise of incredible health benefits. So how does protein folding work? Why does it matter? And how can AI improve our health? That's what we'll explore in this episode of Health Reframed. My name is Jack Snyder. I have a background in biomedical sciences and anatomy and physiology. Furthermore, I've worked in medicine for around 4 years now as an EMT in multiple settings. In this show, I break down the science of medicine and give evidence-based techniques to take agency over your own health. Let's dive into the fascinating world of protein folding and what AI means for the future of medicine. Proteins are what we are made of, by and large. They do virtually everything in the body and are like microscopic tools inside of your cells. But what exactly are proteins? How are they made? And what is all this about protein folding? Alas, to answer these questions properly, I do need to take you through the basics of molecular biology. Now, don't leave. I know this sounds dense and complicated, and some of it is, but I think you need to understand at least the basics of it to give you an idea of the process and maybe an appreciation for the advancements that we have made in recent years. So let's break this down. Proteins lie at the core of what is called the central dogma of molecular biology. This dogma states that DNA codes for RNA, and in turn, RNA codes for and creates proteins. In effect, it explains how DNA, the genetic code of your body, is turned into proteins. And this process is the core process of how life forms and continues. So let's look at this process a little bit more in depth and break it down for the layperson. So let's paint a broad picture. DNA is your body's blueprint. It has all of the information on how to make your body. And for DNA to be useful, the blueprint must first be converted into RNA, which can be used by the cells. This conversion of DNA to RNA is called transcription, since you are literally transcribing the DNA into RNA, like how your phone transcribes your voice into text. In the cells, the voice, in this example, isn't particularly useful to do stuff, to make the proteins. It needs a text version, if you will, to do this, and that's going to be the RNA. Next, the RNA goes out into the cell, and because it contains the instructions on how to build proteins, your cells use the RNA to do just that. This process is called translation, since it's translating what the RNA is saying, in effect, or what is written down in the RNA, into a working molecule, a protein. That's the bird's eye view, but let's break it down a little bit more, step by step. Let's start with DNA. DNA is deoxyribonucleic acid, and it's the blueprint of your whole body. Everything in your body, all of the proteins, all of your organs, it's all stored in the DNA. DNA contains all the information that is required to create you, from your eye color to your kidney to the smallest of proteins inside of your cell. It's all inside the DNA. But what exactly is DNA? After all, we hear a lot about it in genetics, but there's often a black box around the specifics of DNA and how it works. DNA, in a word, is a very, very, very long list of instructions. DNA consists of 2 chains, so you can think of it like winding together 2 bits of string. However, this string is so small that it is invisible to the naked eye unless a lot of it is bunched together. These chains are made of long strings of nucleotides. These are chemicals that encode everything in your body. And they encode this information in a language of sorts. This language is made up of only 4 letters, the nucleotides. And these are adenine, guanine, cytosine, and thymine. But they're often abbreviated to the one-letter abbreviations A, G, C, and T. These molecules link together to form these long chains, and these chains then twist together, forming the iconic double helix shape that many of you may be familiar with. And each chain is said to be complementary to the other, and that means they match together. They click together, almost. And this is because the four nucleotides like to match up. G will go with C and T with A. And to paint that better, it's almost like they're puzzle pieces that fit together perfectly. G and C are puzzle pieces that click together, and so do T and A. So, for instance, if you have a strand of DNA and its nucleotide sequence is AAAGGG, then the other strand that it is wound up to be, or wound up with, is going to be TTTCCC. And the reason for this is because they click together, they actually form chemical bonds, so they chemically connect with each other. On each of these strands lie segments of DNA with specific sequences of nucleotides, and these segments are what we call genes. So each gene is a section of DNA that codes for something in the body. It's almost like a whole sentence or a whole paragraph in the DNA, the language of DNA. So you're going to have a gene that determines your eye color, as well as a gene that may make you more or less susceptible to having cancer. In other words, a gene is a small segment of DNA that holds the instructions for doing one particular thing. Your DNA is stored in the nucleus of your cells in 23 pairs of chromosomes. And all that a chromosome is a really, really long strand of DNA that is folded up and made into a really, really small area. And each chromosome contains many, many genes, and it's all very compressed. All of your DNA together, strung out, would be over 2 meters long, so that's like over 6 feet long. Although the DNA has the instructions, it cannot actually execute the instructions by itself. It's like the master copy that your body uses to refer to. It needs help and it needs machinery. To use an analogy, the DNA is like the foreman of a job site. He's going to have the blueprint for the whole construction project, but that doesn't mean he knows how to build the whole thing alone, and not like he could, he needs a lot of help. He needs workers as well as machinery to build it, and he may even need some people to take a look at the schematics and are more specialized and can say, okay, this part needs to be done this way, this part needs to be done this way, because they're more specialized. They know what to do. And your body is no different. And the first worker that we will talk about here is called messenger RNA, or mRNA. And this is like the foreman scribe. mRNA, as the name implies, is a molecule in the cells that is a messenger. mRNA is very similar to DNA. It is a strand of nucleotides. It is somewhat different chemically speaking, but practically speaking, it can move around the cell and actually do things, which DNA cannot. DNA is confined to the nucleus and can't leave. Whereas the RNA, the mRNA, can leave the nucleus and go out to the rest of the cell. And proteins aren't made in the nucleus. They're made outside of the nucleus, so that's kind of why this is important here. And an mRNA is a copy of genes from the DNA, and it can be used by the cell. But how does this copying happen? Like I said earlier, DNA is normally very tightly bound in the cells to conserve space. Remember how I said that it was over 2 meters long? It takes some serious gymnastics for your cells to contain all that DNA, so it has to be compressed. Think about how you can vacuum pack your clothes or food when you're moving or meal prepping to save space. However, to read the DNA, you do need to open the vacuum bag, so to speak. And this is what happens in the cells. The DNA loosens up and the strands separate. Remember that DNA is made of 2 complementary strands, like 2 pieces of string twisted together, and on these strands are the nucleotides. But the genes exist on each one individually, each strand individually. So once the DNA is loosened and separated, we can begin making the RNA. This process is admittedly extremely complicated, but let's use another analogy to make it more understandable. Think about using voice texting. When you speak the words aloud, your phone processes this and then types the words on your phone. Your spoken word is like the DNA. It contains the original information, but it's not very useful yet, as you're texting your friend who's 100 miles away. They can't hear you, right? Your phone encompasses a process that we call transcription. So you speak into your phone and it turns it into text. That black box, if you will, is transcription in the cell. Transcription is the process by which your cells convert genes or short segments of DNA into RNA. And the main technique, the main machine used for transcription is called RNA polymerase, which reads the DNA and spits out RNA. It creates the RNA in real time. And finally, the text message in our analogy is the RNA. This new strand of mRNA contains the instructions for a protein. This is great news, and we are one step closer. However, how do these instructions work? As we talked about earlier, RNA is made up of nucleotides just like DNA. These 4 nucleotides are adenine, guanine, cytosine, and thymine, or AGCT in DNA. And in RNA, the thymine is replaced by uracil. This isn't particularly important at this level, but I thought I would bring it up. Furthermore, unlike DNA, RNA exists only as one strand, so it would be one piece of string as opposed to 2 pieces of string twisted together. So this helps allow it to be used, and it means that mRNA is a single strand of A, G, C, and U nucleotides in a string. And these letters aren't arranged randomly. They're arranged in groups of 3 called codons. Having these groups of 3 in effect expands the things you can say in the language of DNA. Instead of 4 letters, you now have 64 distinct codons that hold meaning. But what does each codon mean? Some indicate the start of a gene, and this is important because when we actually start creating proteins a little bit down the road here, we need to know where on the mRNA to start making the protein. In other words, where on the mRNA does the code start to have meaning? And some indicate the end of the gene for the same reason. But most indicate an amino acid. For instance, a codon that has the nucleotides UUU, so think of on a string, you have these chemicals, the 3 uracils in a row. This is going to tell the cell that it needs to create a phenylalanine amino acid. And it will do this while the protein is being made. mRNA is a long string of these codons, so a long string of groups of 3. And this string can then go out into the cell, and the other piece of machinery that we need are called ribosomes. Ribosomes take this string and turn it into a protein. They read the mRNA, they see these groups of 3, and then they start creating a chain of amino acids. They connect the chain of amino acids together. So earlier we had the UUU coding for phenylalanine. It'll pluck a phenylalanine from the cell, add it to the chain, and then the next one might code for a different amino acid, and then it'll connect that to the phenylalanine, and so on and so forth. These ribosomes are really the key players in the process of translation. And translation is the final step before getting the proteins, and it is rather involved, similar to transcription, so we don't have to get too far into it here. But in a word, the ribosomes read the RNA and link together a long chain of amino acids, and it ends up being like pearls on a string, almost. So what are amino acids? Amino acids are the molecules that make up your proteins. Yes, we finally made it to proteins. There are 22 amino acids that your body uses to make up proteins, and stringing these 22 amino acids together in different ways creates proteins. However, these strings aren't really useful as strings. Think of proteins as tools, as we said earlier. If you just have a bunch of strings that are containing different materials, they're effectively all the same thing. And a common example to explain the next step is origami. So a string of proteins, a lot like a piece of paper, is pretty unremarkable by itself. However, when you fold the piece of paper, you can have a swan or a box or any number of things. And the long string of amino acids in your cells is going to fold in a very similar way as a piece of paper for origami. And this step is really critical for the function. For instance, your cells have pores in their membranes. And these pores, unsurprisingly, look like little holes, and they are made of proteins. So if you think about the origami example, the piece of paper, that's not going to do much on its own. If you need a pore, if you need a hole in the cell to let stuff in, a piece of paper isn't going to help you. But if you fold the piece of paper into a tube and then you stick it into the membrane of the cell, then boom, there's your hole. And it's similar with proteins. Again, it's very complicated when you get into the biochemistry, but in a word or from a big picture point of view, that's effectively what we're doing. And the process of protein folding is in large part a mystery. And this is where we hit the critical point of the episode, because protein folding, it's really complicated, and proteins could theoretically fold in infinite number of ways. So we have our 22 amino acids, and you can arrange these in really any way you want. You could have proteins that are 100 amino acids long. You could have proteins that are thousands of amino acids long. And different arrangements of proteins different arrangements of amino acids are going to fold in different ways. And we don't really know how. It happens very rapidly, and again, it involves physics and chemistry, and it's really important because knowing how proteins fold can take us from, okay, you have this string of amino acids, what does that turn into? And this is important because we know what the strings of amino acids are going to be in the cells because we know the DNA. We know what proteins are coded for based off of the DNA in the cell, but we don't necessarily know what these proteins do or what they look like or where they're even sent to in the cell. So knowing how to take the string of amino acids and finding out what shape they're going to take really is a paradigm shift. To use another example, it would be like finding a saw and making an educated guess that it's used to cut through stuff. If we can figure out what shape the proteins are going to be, we can make educated guesses on what they're going to do. And that's not all. There's going to be a lot more practical things that we can do with this that we'll talk about in a little bit. And there's been a long disconnect in biology here. We've been able to read the DNA and know the genes for quite a long time now, decades. But we can find the genes and tell you what amino acid sequence the protein that gene codes for. However, we have to spend a lot of time finding out what the protein will look like. The first protein whose shape was found was myoglobin in 1958 by John Kendrew. However, it took him and his team a very long time, and on top of that, they had to use whale myoglobin because it was so much larger. Today, matters have improved, but it still takes a very long time, and it's very labor-intensive to find the shape of proteins, and that is until very recently. Previously, researchers would have to spend long periods of time and money finding the molecular structures of proteins. They did this with techniques like X-ray crystallography or nuclear magnetic resonance spectroscopy. They sound complicated and expensive because they are. And we need not delve into these today, but again, the names alone indicate how complex they are. However, for the past 30 years, researchers have been trying to model protein folding digitally on computers, so that in theory, we could input an amino acid sequence and have the software reliably spit out the final structure. Since 1994, the Critical Assessment of Structure Prediction, or CASP, has undertaken a worldwide collaborative effort to make such a software. Every 2 years, contestants will put their software to the test. The organizers will give the contestants amino acid sequences of proteins where the structure is known, and they will have to use their software to find the final shape. In other words, they give them the building blocks, and they have to use them to build what the body builds. It would be like giving someone all the materials to build a house, but the only resource they had to build it was simply by looking at other houses and seeing how they were built. For the first 13 iterations of the competition, contestants were coming up pretty short. CASP had stated that if someone could create a software that was 90% accurate in finding the shape of a protein, that would be sufficient. That's their gold standard number. But year after year, the best that contestants could do was nowhere near this number, 50%, 52%, 55%, 60%. All of that was until 2020. AlphaFold 2 is a protein folding modeler that was created by DeepMind, which is a British-American machine learning AI company run by Google. AlphaFold 2 really swept the competition in 2020, and more importantly, it reached that 90% mark. And experimentally, in the past decades of using lab techniques to find protein structure, we've found around 200,000 proteins. But with AlphaFold 2, and other new emerging technologies, we are now able to predict the structure of over 200 million proteins. This is huge. This is a 100-fold increase in knowledge that we gained instantly, 2 orders of magnitude. But how does AlphaFold 2 do this? The recent boom in AI can largely be attributed to advances in machine learning. Machine learning involves giving software copious amounts of information and then training it on that information. For instance, many of you will have used ChatGPT, and ChatGPT is able to create sentences and write papers not because it knows grammar or knows what to say, but because it has learned so much information that it can accurately predict what the next words will be. So if you ask it to write a diss track in the style of Ryan Reynolds, it's going to analyze everything that Ryan Reynolds has ever said and then just predict the most probable string of words based off of your prompt. And this is pretty much how the protein AI models work. Instead of being trained on spoken language, they're trained on what is called the protein database. And what this is a database that contains all of the structures of the proteins that we have found. That's that 200,000 number from earlier. The AI looks at all of these structures and then uses them, as well as physical constraints, you can't defy the laws of physics, really, to spit out a prediction of the given string of amino acids. And this is a very simplified explanation, given that AlphaFold 2 uses really complex machine learning, but it gets the idea across for our purposes. If you want to learn more in depth, Veritasium has a phenomenal video about this that I'll link down below. So this is all well and good, but what does it mean for us? What will we be able to do in the upcoming years as this technology progresses, and how are we going to use it? Furthermore, how should we use it? The leap forward that AI has given us in the world of protein folding cannot be overemphasized. Again, this leap puts us 3 orders of magnitude ahead of where we were previously. To put that into perspective, 200,000 seconds is 2 days, 8 hours. 200 million seconds, on the other hand, is 6 years and 4 months. In other words, this is a huge breakthrough. And now that we have all this information, what are we going to do with it? After all, information for information's sake isn't particularly useful to us day to day. To start, let's look at a paper by Yang and colleagues, where they list some of the main areas in which AlphaFold and other softwares will be used. These include structural biology, drug discovery, protein design, target prediction, protein function prediction, protein-protein interaction, and biological mechanism of action, to name a few. The first of these, structural biology, is perhaps the most important. A large part of molecular biology for the past few decades has been focused on finding the structures of proteins experimentally. This entire field is going to need to adapt to these changes and use AI to expedite their research. Though it may seem like AI may impact jobs in this area, it also gives a lot of opportunity for researchers to simply amp up their research and make more discoveries more rapidly. These discoveries will help fuel much of our understanding of how the body works, as well as how some of the invaders of our body work, like viruses or bacteria. And these findings can help assist in the next exciting area that AI is going to impact, drugs. One could imagine that we could find a new protein from bacterial DNA. These proteins could then be targets for antibiotics, as this is how antibiotics work. They target normally a protein or something that only the bacteria has. It kills the bacteria and not us. Furthermore, research has been done in the field of cancer. Being able to find places for drugs to actually interact with cancer may put us one step closer to finding more effective treatments. Furthermore, being able to sequence the genetic code of cancer and provide personalized drugs to treat your cancer specifically might be possible. Really, the possibilities seem endless in terms of drugs and personalized medicine. Think about it. If we were able to sequence your entire genome and then design unique treatments for you based off of the proteins in your body, the sky really does seem the limit. Now, naturally, I would imagine many of you will have raised your eyebrow at that, because this does raise some serious ethical questions that we're going to address later, so hang tight for that. Beyond discovering proteins that are inherent to the human body or bacteria, AlphaFold gives us the opportunity to create proteins, create new and unique proteins. The same paper by Yang and colleagues outright says that we're going to enter into a new era of protein design. This is because AI protein software removes obstacles that have long plagued the field of protein design, like predicting the structure from an amino acid sequence. These programs can do this task with relative ease, making it a moot point. One application that I can imagine here is antibiotics, like I said earlier. In the past, we have been able to give monoclonal antibodies to sick patients to help them get better. For instance, during COVID, for a period of time, you could get monoclonal antibodies. These antibodies give your immune system a head start in targeting the virus. However, making these antibodies is a very involved process that isn't always effective. However, I could imagine where we could do a test on a patient, and then based off of the DNA of the virus, design and manufacture antibodies using AI that could be given to the patient. Or even more general, we could create these antibodies at mass scale during the cold and flu season to help people get better quicker, almost like an improved flu shot. Using AlphaFold to understand how the body works opens more doors as well. As we come to a better understanding of the structure of the cells in your body and all of the proteins in them, we will be able to make better predictions about what kinds of things you may be more susceptible to and how to design treatments and drugs that are far more specific, doing exactly what we want them to do and nothing more. Here is an example. Many psychiatric drugs at the moment have very broad effects. SSRIs, or selective serotonin reuptake inhibitors, are not very selective, despite the name, meaning that they impact many areas that serotonin has effects in the brain. Serotonin has effects all over the brain. However, if we are able to better sequence these serotonin receptors and where they are in the brain, then we may be able to make more specific drugs that only target the specific receptors that we want in the brain. And this is going to help reduce side effects and really just hone in on doing exactly what you want the drug to do. In a similar vein, we are also going to make a lot of progress in determining the actual function of proteins. After all, like I said, if we know the structure, this is often half the battle when it comes to the function of proteins. Proteins are tools, and more often than not, their structure hints at their function. Real quick here, I want to talk about one protein that's really awesome and cool, and it's called ATP synthase. ATP is the protein that creates the bulk of ATP in your body. ATP is the energy currency. It stores energy that your body can use. And the way it does this is really cool. So it's like a water wheel. The mitochondria move hydrogen ions inside one side of the membrane. And this means that there's a lot of hydrogen ions outside of the mitochondria and almost none inside. And because of some chemistry, the hydrogen ions want to move back inside to balance that out. Think of if you move water to a high area and then you punch a hole in the bottom of a box that the water is filled with, the water is going to fall out to evenly disperse. It's almost like this. So what ATP synthase does is it allows the hydrogen ions to move back inside. And in doing so, it spins. It uses the hydrogen ions to spin the protein, pretty much like a water wheel. And this spinning motion effectively gains the energy needed to make ATP. So instead of water moving the wheel, it's hydrogen ions. And the energy that this provides is used to create ATP. This is very cool, if you ask me. And the reason I bring this up is because it shows how if we know the structure of a protein, we can start to extrapolate function. If it looks like a water wheel, then maybe it is one, so to speak. And these are just some of the applications of AlphaFold and AI for protein folding. Others would include vaccine design, treatment of rare diseases, and finding the effects of mutations in the genetic code. AlphaFold is really exciting and really groundbreaking. However, it does have some limitations. Primarily, it suffers from the problem that all large language models, like ChatGPT, have. And that is they're only as good as the data that they're trained on. AlphaFold was trained on the protein database, and as a consequence, it can only be as good as the database. One of the key points here is that the protein database consists of structures that were found in a lab. This isn't a bad thing, but it is important to understand that no matter how hard we try, the lab isn't the body. A protein may fold one way in the lab, but a different way in the body. For instance, knowing this limitation as well as others is important when we use these programs. That said, though, the protein database is indeed the best we have, and it is very robust, so I'm not trying to knock it. I'm just adding a little bit of nuance here. So it's an excellent place to start. And the limits of AlphaFold raise a couple of other questions as well, and these are more ethical and moral. How should we use it? What ethical considerations should we take? And are we getting dangerously close to losing our humanity with some of these breakthroughs? So this is really the nexus of all of the AI conversations and kind of where they converge, because it's important. We're playing with some powerful things here, and we really need to sit down and think about how do we use them? How should we use them? And what are the guardrails that we need to put in place? So any technological advancement is going to come with these ethical and moral considerations, as well as a lot of uncertainty. During the Industrial Revolution, many people were terrified that these new machines would take away their jobs. And the machines, they were faster, they were better, and they were cheaper, so they did take away some jobs. However, as we all know, the Industrial Revolution provided a lot of new jobs. It took away some, but then it provided different ones, often ones that weren't as labor intensive and not as dangerous. And the new technology provided many opportunities for people to have factory jobs, for better or worse. And AI seems to be following the same path, especially in the field of protein folding. I'm optimistic that we may be going through a similar societal change. Will AI replace many of the jobs that people do currently? Maybe, probably, but it's also going to open a lot of new opportunities for jobs that never existed before. To give an example, I've applied to a couple of these AI jobs that are new and didn't exist before. What they are pretty much is an AI fact checker, where you go in and fact check what the AI is saying with your human knowledge that is oftentimes a little bit more nuanced and able to catch out some of these details that AI misses. And these are jobs that really never existed before, but now they're a growing market, and I'm assuming it's going to continue to grow. And this is just one example of many that I can think of off the top of my head, but more are inevitably going to come. AI could eliminate the need for labor-intensive protein folding techniques, but that could free up resources for more complex projects. And I think that humans have this virtually limitless capacity to adapt to their surroundings. However, I think that there is some importance of being privy to what unintended or hidden consequences AI may have. The Industrial Revolution caused many more people to move to cities, which had the unintended consequence of people not feeling as in touch with small, tight-knit communities. They didn't touch grass as much. I mean, sure, they had these factory jobs that probably paid better, but there were some trade-offs here. And this is a problem that we still contend with today from the Industrial Revolution, and we're still working for solutions on it. And my point is, we need to be on the lookout for what the equivalent of these consequences would be for AI. And I think that one of the most salient discussions that pertains to AI in biology is, should we be turning over the genetic code and manipulating it with designer compounds? In other words, are we overstepping how much we're manipulating our own biology, given that it seems plausible that in a few years' time, we'll be able to sequence your whole genome and design drugs specifically for you? Are we playing God here? How should we wield the power of genetic engineering? And to add insult to injury, how are we going to make sure that we have the proper cybersecurity to protect your genetic code if it's inevitably going to be stored in a database? And these are like just a few of the questions that we need to answer. And in my humble opinion, I'm not sure we're pacing ourselves properly. Technological advancement trudges onward at lightning speed without giving us much time to answer the simple question. Just because we can, does that mean we should? I think a decent place to start with all these questions is by once again looking back in time. Medical and biological advancements are nothing new, after all. Many of these breakthroughs were initially met with rejection. Some notable examples here would be hand hygiene, vaccines, and the fact that stomach ulcers are caused by bacteria, not stress or spicy food. We now know that all of these advancements have helped save many, many lives and have made the world a better place. So it is clear that sometimes early rejection may be in fact wrong. On the other hand, the inverse is also true. The history of medicine has shown many examples where early acceptance may prove to be deadly, like how doctors used to prescribe and recommend cigarettes. And there's also a bleak history here in terms of drugs. 1 in 3 drugs that have been approved by the FDA get flagged for safety concerns after they've been approved. A notable example here is the drug Vioxx. Vioxx was a drug developed by Merck in the 90s, and it was marketed as a painkiller. However, after it had been approved and was being used, tens of thousands of people were dying from it due to heart attacks and other issues. Merck took the drug off the market, but it was too little and too late. But that's not the worst part. The worst part here is that Merck knew about this. They manipulated the scientific data from their research so that their drug would get approved. And to add just the cherry on top here of this story, Merck still ended up profiting from this drug. I forget the exact numbers, but I believe they made around $7 billion on this drug. And then they got sued in a class action lawsuit for around $4 billion, which at the time was the largest lawsuit in the United States history. So they lost $4 billion from the lawsuit, and then they had to pay, let's call it $1 billion or $2 billion in research, development, marketing. So they still profited here, which not a very good look. And it's one of those parts of history that just kind of makes you cringe, but it's a part that we have to learn from and be aware of. So what I'm trying to say here is history puts us in a bit of a pickle. Over time, medical advancements have caused incredible improvements in health, and at the same time, they've caused catastrophic disasters in health. So what do? First, we need to be attentive and level-headed. Letting ourselves get clouded by bad actors or antiquated beliefs can be detrimental. To give another quick example, many people pushed back against anesthesia when it was first used because they thought that pain was something that wasn't to be tampered with and it was part of the human experience. So in other words, we need to be willing to update our preconceived notions, but only if it is indicated to do so. And at the same time, we need to know when to slow down and make sure that we're not stepping on a landmine. Research is going to be important here. If we're designing drugs based on an individual person's DNA, we want to test this on animals first. I think this is true for anything that's genetics related. I also think that toying with genetics is a very dicey game. From designer babies to total and utter sedation akin to 1984 or Brave New World, it's really not hard to imagine dystopian futures in the not so distant future with AI and genetic engineering. I think something else that we need to keep in mind with all of this is the fact that humans do indeed have agency. Ignoring the fact that people can change their lives for the better may have disastrous consequences. I think that we're seeing this to a certain degree with overprescription of some medications like psychiatric medications, and it will be important going forward to be careful designing drugs, making certain first that there is actually a biological basis for the problem. That's one of my big things on this channel. I try to give all of you the agency and the empowerment to actually take control over your own health, because I think in a lot of cases you can, and I think it's important to do so. And indeed, one of the major concerns with psychiatric medications is that they are used as a band-aid for a problem that the patient may have a decent amount of agency over, as opposed to a correction of faulty biology. So what I'm saying here is that we need to make sure that we're actually designing drugs and using the AI and protein folding to address actual biological issues, not issues that could be fixed with self-improvement or hard work or empowerment or personal growth or whatever you want to call it. Because in my opinion, that just it diminishes from our agency, and that's a big part of our humanity, right? As humans, we have free will and agency to change the world around us and make it better, and I think degrading that is a big blow against our humanity. However, programs like AlphaFold do give us an edge here as well. If we can determine your genetic code and whether or not you have dysfunctional neurological problems, then we may be better equipped to give medications. So at the end of the day, it's a four-dimensional balancing act that's going to require many, many, many people from tipping it over. And there's a lot of nuance, and it's important to have conversations and work these things out and have checks and balances in place so that things don't get too ahead of themselves. But if we are slow and careful with our research, we keep a clear head and are open to the possibility of both good and bad, we may be able to navigate this minefield. I also think that open conversation about AI is vital. We need to make sure that AI is not this entity that is controlled by a particular group of people. It needs to be a collaborative effort. And we also need to nail down questions like, what does it mean to be healthy? How do we make sure AI is oriented towards what is best for humanity? What is best for humanity, right? And this gets into deep philosophical discussions. I think that at a bare minimum, what is best for humanity consists of minimization of unnecessary suffering, promotion of healthy in-person communities, more people living long and healthy lives, just to name a few. But it seems to me that we really need to sit down and redefine what it means to be human and what is the best way to be a human and what's the best for humanity, because I think that's where we often get into trouble with these conversations, is we're not agreeing on these fundamental things. Indeed, these questions lie at the heart of AI, because I would wager that for most of us, save a psychopathic few, think that these questions are important and that training AI to have our best interests as humanity at heart is very important. And once again, I want to stress the importance of conversation. In my experience, no matter how different someone may seem, if you both enter into the conversation with curiosity and respect, you're going to find a lot of common ground. So that's one of my big selling points. Maybe my biggest selling point with this whole segment is go out there, talk to people you wouldn't normally talk to people you disagree with. Be authentic, be curious, be respectful, and no matter who the other person is, I guarantee you there's going to be some common ground. There's going to be a common humanity there that you can find. We are all human after all. And one final thing I want to touch on here is legislation. I am not a lawmaker, but I think it is important to think about what kinds of laws lawmakers should be thinking about for AI. I think that making AI open source is a good place to start. Like I said earlier, making sure that this is a little bit more decentralized and not held in a small group of people is important. That way, the power is not concentrated, and limiting the uses of AI until they're proven, especially when you combine it with genetics, could also be important. I think really what legislation here needs to be is a buffer to prevent AI from running out of hand, as well as making sure that it is something that is open to everyone. Like I said earlier, technology tends to progress a lot faster than the ethical and moral conversations that we have. And I think that's where legislation really has its role, is it needs to slow the progress just a little bit so that we can have the time to reflect on what we're doing before it gets out of hand. AI in protein folding represents a massive leap forward for medicine, but it also raises complex ethical questions. Just because we can design drugs tailored to your DNA, does that mean we should? Navigating this is going to require open, curious, and respectful conversations that value all opinions, careful research, and then a shared understanding of what it means to be human. Furthermore, it will be paramount to heed the lessons from the past and really try not to make the same mistakes, as difficult as that may be. And that starts with asking the hard questions and working them out together. Molecular biology, artificial intelligence, convoluted ethical dilemmas. We truly are living in the future, guys. Although the advent of AI and everything that it is rapidly becoming capable of may be daunting and frightening, but I think that its use in biology gives an important window for us to look through. It shows us that if we can properly harness this technology, AI can be an unstoppable force for good. AI is already pushing molecular biology to new heights, and the research thus far gives us a lot to be excited about. Personalized medicine, a better understanding of our own biology, and much more. However, as cliche as it does sound, with great power does indeed come great responsibility. And AI's rise marks a paradigm shift in society and technology and the world and everything else. And on the scale of the printing press or even the atomic bomb. What we will do with this power is surely going to shape our future. Every technology has the capacity for both good and bad, and our job is to be the stewards that guide this new world and the new technology along the best course. And perhaps AI will help us do this with much more precision, grace, and efficiency than ever before. In conversations like this, where we try to approach the technology with nuance and understanding, is vital. So go out there, have this conversation. Share your viewpoints, hear other people's viewpoints, and just keep talking. Educate yourself, because it's really going to take all of us knowing what's going on to reach a conclusion and having these discussions. All of that said, I am hopeful and super excited to see what researchers are going to do with AlphaFold 2. I think that we're in store for a wild and exciting future with endless possibilities.